Hello 8th graders, this video is to introduce you to the procedures part of the science fair. So this is the same document that has the prediction information on it, but as I said in the last video, if you are starting this step at the procedure step before you have an approved idea, you need to stop and go back and get your idea approved. If you start working on this and it turns out that you can't do the idea that you've picked, you may have to restart completely, and this will be all wasted effort, which would be a sad thing. So make sure you have an approved idea. You will know by going to the bottom of your rubric from the project idea form, and if you have the box filled in that says number 2, approved science idea is approved, then you can go on. So I'm working from the assumption that you have an approved idea for the rest of this video. For the procedures, the most important part of your procedure, or that you know about the procedure, is that it's going to come in 3 parts, and it has a giant portion of your grade for the final. So remember, science fair is your final score. The procedure is going to be 20% of your entire grade. So that's the difference between a low B and a high A. That's like almost 2 whole letter grades. So this is an important part. I believe you can do this. I've seen your procedures from other parts of science class, and you're going to do great. But here are some things to recognize. If you have a highlighter or a pencil or pen, please be marking along with me as I point out some of the things on your paper. So the first part I want to talk about is the materials. You're going to have a section called materials, and in this section, please make sure that you are listing all of the amounts. So if you say that you want the person to get some water or some salt or blocks of wood or something, you need to say how much of that ingredient or how much of the ingredient they'll need for the entire experiment. All of your materials, if they are generic, things like water or sand or paper cups, if it doesn't matter what brand they are, then don't list it. If you use Dixie brand cups for your experiment, but it doesn't matter that the person uses Dixie brand cups, then just say paper cups. That's totally cool. But if, for example, you were going to do, let's say, an experiment on candles and how fast they burn, or something about how they burn. Well, if somebody was going to try to do your same experiment and used a different brand of candle, then it's probably likely that they're going to get a different result. So in that case, the brand would matter. So think about, how am I going to make sure that if somebody does my exact procedure, they're going to get the exact same outcome as long as they follow my steps? If the brand matters, then you need to list the brand. If it's not that big a deal, like for example the water thing, water is water, and that won't matter that much. Don't list anything unnecessary. So like, if you're writing in pencil so that you can record your data, that is an unnecessary material in order to gather your data. That's not required. So make sure that that gets taken out of there. Materials list should just include the things required to collect your data. The next section is going to be the actual procedure, so the step-by-step process through which somebody would have to go to gather your data. So some of the things to point out here is that these need to be numbered steps. So the reason why they're numbered is because there's an order implied. First you do this, second you do this, etc., etc. The materials list is just a list, just bullets, kind of like the way this paper looks with bullets. No numbers in the materials list, yes numbers in the procedure. You need to explain in the procedure like what should get recorded. So there are a lot of observations that could get made throughout a science fair experiment, but I need you to say in the procedure what specifically should be recorded. So if it's a mass, say record mass at a certain point, or if it's a distance, record the distance of in your procedure, something like that. You also need to be able to, or you need to say how many trials. The requirement for this assignment is at least 10, but that can change a little bit if you're doing animal or human trials. If you're doing animal or human trials, it should be probably at least 3 times that many, so 30 trials. The reason is because it's hard to control for animals and humans, so we have to get over that problem by doing way more experiments. Now, one of the things that gets mistaken here is what is a trial and what's an experimental group. So I wanted to share an example with you. Let's say that you're going to test how much plants will grow as you shine different types of light on them. So you're going to use like a heat lamp and a fluorescent lamp and a bunch of different lamps, and you're going to measure how much they grow. Well, in that kind of an experiment, your experimental groups are the lights. So for example, you'll have one group that's going to get fluorescent, one group that's going to get heat lamps, one group that's going to get some other third type. Trials means that you're going to have 10 plants in each group. So you're going to have 10 plants that gets the fluorescent light, 10 plants that get the heat lamp, 10 plants that get the incandescent, whatever. So if you had 3 experimental groups with 10 trials per group, that's 30 trials. So keep that in mind as you're writing your trials. The last thing that I want to point out in this section is the control variables must be in your procedure. That's often kind of confusing for people because they think, well, yeah, I just listed my control variables at the end, what I'm going to control for. But that's not good enough. They need to show up in your steps. So an example of that would be if the amount of, let's go back to the plant experiment, and you're going to shine different lights on it. If you've decided that it's important to control for how much water each plant gets, that's a great control variable. Well, somewhere in your procedure it should say how much water to water each plant with. You can't just say water the plants. If it's also important that it gets watered at the same time of day, well, then you have to say what time of day and how much water, and as specific as you need to be. So all of your control variables should show up in the way that you write your steps. The last section, section C, is for your variables. You're going to have 3 little sections, one that says your IV, your DV, and then a list of your control variables. It's just a list. I'll show you an example in a second. Your independent and dependent variable must match your hypothesis in the exact same wording. It's going to become pretty important because it's embarrassing when you're presenting and your hypothesis and your procedure don't match. The judges are going to say, well, which experiment did you do? The one that your hypothesis says you did, or the one your procedure says you did? And you want to avoid that embarrassment. Also, it says at least 3 meaningful CVs. That's for like the most simple science fair project possible. You should probably have, like, the average is somewhere in the neighborhood of like 6 to 10. More is always better with CVs, but on the rubric, 3 is the minimum. So here's some exemplars. Be sure that you read this. I put the statement of the problem on here so you understand what the example is all about. So there's a prediction that was in the last video. Please notice for this exemplar procedure that they have amounts in metric for every single ingredient that they're going to use, and things that don't matter, things like a permanent marker, which you might think, hey, he said don't put pencil. They actually use the marker to mark their groups and to label. So it doesn't matter what brand it was, so it's not branded. But all the amounts are there. For their procedure, notice that they've got them in numerical order. Their first step does not say gather materials. That's a huge one. To a scientist, that's what this part means. Part A, where it shows the materials list, you're basically saying go get this stuff. Don't add it to your procedure also. It makes you look silly. And then one of the coolest things about this procedure, it makes it really good, is that they use some steps that say repeat this a certain amount of times. Anytime you can make your procedure shorter and more concise, that means saying the same thing using less words, you should do that. So anywhere where you can say repeat these steps but do it this way, or just repeat the steps to get your trials, that's where they're including the trials by saying 10 times. That's always better. And then for their variables section, they have listed their variables. So at this point, you need to go off and start working on your procedures, and don't forget to click turn in when you're done. Good luck.